All right, so marker-assisted breeding, that's a very important application of markers. There's another application that we can use, and we've been using it in Australia on a number of occasions, and that is to, what I've put here, deal with suspected cases of virulence for resistance genes. Okay, so this gene SR26, it's effective against UG99. In fact, there's been no virulence reported anywhere in the world for SR26. We've used it in Australia in at least 30 wheat cultivars since 1971. It was originally found by this guy here, Doug Knott, in Canada. It's from Thinopyron ponticum. So Doug actually did an intercross and he introgressed this chunk of Thinopyron ponticum DNA into wheat, and it carries this all-important SR26 resistance gene. You can see what it looks like here. All of these wheat lines here carry SR26, sort of what I would call a 1 or 1,2 minus infection type. Really important resistance gene for us in Australia. Back in 2010, I had a phone call from a colleague of mine in South Australia. He said, we've got a farmer in South Australia growing this variety kite that carries SR26, and this is what his crop looks like. So straight away we were worried that we'd had a mutation in the rust pathogen for virulence for SR26. And we were quite confident that the variety he was growing was kite because it was awnless. We only have 2 or 3 awnless spring wheats in Australia, so we were quite confident it was not a case of wrong cultivar identification. There was a lot of stem rust in there. We were worried we had a mutation and we had a new pathotype that took out SR26. So the obvious thing to do was to take a rust sample and test it and work out what the pathotype was and determine whether it was virulent. But that was going to take 2 to 3 weeks. So there was another thing we did that gave us a pretty quick answer. We had a marker for SR26. So the guy went into the crop and he collected some leaves of infected plants. We extracted the DNA, and this work was done down in South Australia by my colleague Ian Dundas. And Ian ran, in fact there was also this variety Blade being grown by the farmer. He ran seed DNA from what is it now, 9 plants or seeds that he took off Blade. He actually took seeds, okay? And 5 off kite. These are the positive controls that he put in there. Clearly you can see none of those plants from the farmer's field carried SR26. So this then allowed us to say, well, thank God for that. Whatever it is he's growing doesn't carry SR26. So we probably don't have virulence for SR26. Had they all been positive, it would have been very, very suspicious. We would have been extremely worried that we had virulence for SR26. And as it turned out, when I did the pathotype identification 2 weeks later, I was able to say no virulence for SR26. All right, but here we were able to use the marker for SR26 to assess the possibility of the presence of that resistance gene in the farmer's crop. He was growing these 2 varieties, and that came back negative within 24 hours and told us that whatever it was he was growing didn't carry that resistance gene. And we're now doing this in the...
No, but 1 minute. Number 18 doesn't carry a very big
right.
band.
Yep.
In number 18.
Yeah, it doesn't. It's positive, but it's very, very faint. So this is probably not very good quality DNA here. But clearly Blade, there were 2 blades. This one is actually a new translocation line that Ian's produced that carries SR26. Here we have Westonia plus SR26, Kite again, and Westonia plus SR26 here is duplicated. So there's 2 there. But they're all positive. All of the samples from the farmer's paddock, negative. So didn't carry SR26. There's another dimension to that story I won't go into that's rather interesting, but anyway, we were able to answer the question very quickly that the variety did not carry SR26, and of course the pathotype analysis told us 2 weeks later that in fact the variety, that the pathotype was avirulent for that gene.